So I'm delighted to introduce Stephen, who is a director of the Protein Data Bank and founding director of the Institute for Quantitative Biomedicine at Rutgers. And he will tell you about the Nsp5 main protease. Over to you, Stephen.
Thank you very much, Sagar, and my thanks again to all of the participants for joining us today. My subject is SARS-CoV-2 Nsp5, otherwise known as the main protease. My talk will begin by introducing the enzyme and its role in the life cycle, and then I'll go on to talk about the enzymology and the structural biology, and then summarize the analysis results that we produced looking at the evolution of Nsp5 during the pandemic. I'll touch briefly on the impact of the Protein Data Bank on US FDA drug approvals to set the stage for what will be, I apologize in advance, a sobering postscript, and I'll leave you with, I think, a valuable insight into the importance of a robust planning for public health emergencies. And then, of course, I'll thank everyone who contributed to this work. So you've seen a variant of this slide before. The Nsp5 main protease is one of the two proteases responsible for cutting the polyprotein. So you can see that the SARS-CoV-2 genome, when it arrives in the cell, looks just like a messenger RNA. It has a cap and a poly A tail. These two open reading frames, 1A and 1A, 1B, generate the polyproteins shown here. Then there's a series of subgenomic RNAs, which are actually translated at a different time in the virus life cycle, and they include the spike protein, about which you'll hear more from John Williams, the E protein from Grace Brannigan, and a bunch of ORFs and the M protein and the nucleocapsid protein. So returning to the polyproteins, you saw that the PL proteinase, the papain-like protease, cuts at these 3 sites, and then the remaining 10 sites in the polyprotein liberating the residual non-structural proteins are cleaved by Nsp5, otherwise known as the main protease or the 3CL protease. Nsp5 is a 306 residue globular cytoplasmic protein. It functions as an obligate homodimer, as you can see here in this preview of the structure. As it's expressed as part of polyproteins 1A and 1B, we're left with the question of how it actually excises itself. And we know from work done on the SARS Nsp5 that the polyprotein itself dimerizes through through 2 copies of Nsp5. And in this so-called immature dimer form, it's able to excise itself from the polyprotein. And then it goes on to release non-structural proteins for itself and NSPs 6, 7, 8, 9, 10, 12, 13, 14, 15, and 16 from the polyprotein. There are 2 active sites, one here and one here in this attractive drug discovery target. And occupying the active sites in this particular view are bound substrate analogs shown in pink. This is a chymotrypsin-like enzyme, hence the moniker 3CL. It's a member of the cysteine endopeptidase family with enzyme commission number EC 3.4.22.69, which means that it uses a cis-his catalytic dyad, which is here in this ribbon representation of the protein, to cleave sites resembling the sequence shown here at the position of the slash. The catalysis proceeds in a number of steps, one of which involves the formation of a so-called acyl intermediate, wherein part of the substrate is covalently bound to the active site cysteine prior to subsequent release of the second half of the substrate to generate the full product, the 2 halves of the 2 portions of the polypeptide chain that get cleaved. This particular inhibitor is a substrate analog that remains covalently bound in the active site. It doesn't undergo the so-called deacylation step. This is moving on to the structural biology. Nsp5 is a two-domain protein. The upper N-terminal domain, sorry, the lower N-terminal domain here is largely alpha helical, and this is responsible for the dimerization of the enzyme. The upper C-terminal portion of the enzyme, the C-terminal domain, is largely beta-sheeted structure, a couple of short alpha helices here and here, and this supports all of the catalytic events. There are now approximately 200 structures of SARS-CoV-2 main proteases, Nsp5s, in the PDB. The very first of these, PDB ID 6 Lima uniform 7 using the NATO alphabet, was released on the 5th of February 2020. And I want you to reflect on that for a moment, because the sequence of the virus was not publicly released until about the 10th of January. So in a matter of just over 3 weeks, a team at Shanghai Tech went from a synthesized gene, knowing the sequence of the genome, to an express protein, to crystals, to a three-dimensional structure with this substrate analog bound in the active site. That particular group at Shanghai Tech was able to move so quickly because they had previously determined some structures of the SARS-CoV-2 Nsp5, and I'll return to this at the end of my talk. Among the structures, the 200 or so structures that are in the PDB today, are 116 so-called PanDDA structures that came from the Diamond synchrotron in the UK, located just outside Oxford, where I did my PhD. The team at Diamond used crystallographic screening to look at literally thousands of small molecule compounds. They're called fragments, and to ask whether or not any of these fragments, these subcomponents, chemical moieties that could make up drugs, bound to the enzyme, and they got more than 100 hits, and they've put 116 of these structures into the protein data bank already. Each of these co-crystal structures tells its own story about the chemotype binding properties of the enzyme. And you can see here this small fragment. It's got the halo around it in the active site. This is the active site sulfhydryl, to which you saw the substrate analog covalently bound a moment ago. So this is a non-covalently acting fragment, sitting in exactly the same position that one would find a substrate analog and one would find substrate. So these provide the information in these 100+ structures provide important clues as to opportunities for chemical synthesis of potent selective enzyme inhibitors. Continuing with the structural biology of the enzyme, the PDB also contains 35 apoprotein structures of Nsp5, in which they are just crystallization solutes, no ligands per se, and then another 50 structures of Nsp5 with bound drugs, with inhibitors, and a variety of peptide-like molecules. And I just highlight 4 of these cases for you. In order to understand whether or not drugs could be repurposed from treating one disease to treating the SARS-CoV-2 infection, a considerable amount of work was done crystallographically to look at previously approved US FDA drugs against a variety of viral targets. So there's a structure of boceprevir, a hepatitis C drug bound to Nsp5, the structure of telaprevir, another hepatitis C drug inhibiting the hepatitis C viral protease, is depicted here again with this sort of halo representation. It's covalently linked again to the active site sulfhydryl right here. Here's the other half of the catalytic dyad, the active site histidine. And again, not surprisingly, there's another, excuse me, there's a structure of another hepatitis C virus protease inhibitor nirlaprevir, also in the PDB. What did surprise me when I was examining the list of structures in the PDB that have, of Nsp5 structures in the PDB that have ligands, is there's even an investigational kinase inhibitor, masitinib, bound to the protein. Now, none of these are potent inhibitors of the enzyme. And for that reason, none of these were repurposed as potential therapeutic agents for SARS-CoV-2 infection. But what this collection of approximately 50 structures provides are potential starting points for structure-guided drug discovery. And I'll a little bit later in the talk, I'll actually explain why I think that's so important. Now, moving on to the analysis that Sagar just introduced to you of trying to understand the evolution of the SARS-CoV-2 proteome in 3D during the pandemic, I want to again acknowledge the large number of people in the PDB team and in the Kare Lab team and all of the summer interns, including this very talented student from Watchung Hills Regional High School in Warren, New Jersey, who's actually taking part in the boot camp, in the crash course today. Thank all of them again for the work that they did in analyzing nearly 50,000 viral proteomes and finding that all 29 viral proteins that are encoded within the viral genome underwent amino acid changes during the course of the pandemic, running from December through to almost the end of June 2020, and then contributing to all the efforts involved in doing the structure modeling and doing the energetics calculations. The findings for Nsp5 are summarized here. We analyzed a total of 46,797 viral proteins. 94% of these viral proteomes had Nsp5 completely unchanged. So this is the power of Nsp14, the proofreading enzyme. The 6% or so that had NSP changed, we analyzed these and we found that there were 217 unique sequence variants, 189 of which involve single amino acid changes, 26 double changes, and 2 triple changes. And like the data that Sagar just reviewed with you for the entire set of study proteins and for the PL protease in particular, most of the substitutions that we're seeing are non-conservative, about 60% in this case, with the remaining 40% being conservative. And again, looking at the modeling energetics of these 217 structural models, these variations on the original sequence from Wuhan, China, we can see that only about 13% map to the hydrophobic core, with the other amino acid substitutions being more or less equally split between the boundary layer and the protein surface. Again, and this is not unexpected, this is what is seen in viral replication, is that most of the changes are marginally destabilizing. Interestingly, we did see one very intriguing amino acid substitution in the active site. There's one sequence in which the histidine of this his-cis catalytic dyad has been changed to another amino acid. There's a reciprocal change in the sequence involving a change to a histidine over here. And there's also another histidine over here, which begs the question as to whether or not there's an alternative, probably less efficient catalytic dyad available to the virus. In the lab, for SARS-CoV Nsp5, if you mutate this histidine, the enzyme's dead. So we're still trying to understand what's happening in terms of this change in the catalytic dyad. There are, as Sagar showed for the PL protease, some other changes in the periphery of the active site that could affect inhibitor binding and could be sources of potential resistance in the context of a given therapeutic. The other analysis that was done was to look at the dimer interface. There's a modest number of changes in the dimer interface itself, and we don't believe that those are significantly destabilizing, but it is possible that that other changes on the edge of the dimer interface could affect the stability of the dimer, either increasing it or decreasing it. We really don't know which. So the key lessons learned here from the work that we did in Nsp5 during the course of the summer with the 12 summer interns is that mutations are relatively rare during this 6-month period that we were able to download sequences from GISAID. And we presume, of course, this is the effect of the proofreading exoribonuclease Nsp14. The results confirm that Nsp5, the active site of Nsp5, represents a very attractive drug target because it's so highly conserved. If you put an inhibitor into the active site, you're going to stop the virus cold. The PANDA structures that I summarized, more than 100 of them, tell us about the chemotype binding properties of the active site that helps with making plans for medicinal chemistry. And of course, the co-crystal structures that I highlighted, and there are about 50, also are, will be useful for any group trying to actually do structure-guided drug discovery, whether they're in academe or in industry. And I'll say something now about the impact of PDB structures and structural biologists on US FDA drug approvals, because I really want to make the point very strongly that 3D structural information is central to drug discovery these days. My colleagues and I recently published 2 papers that analyzed in quantitative terms, not anecdotes, but quantitative terms, the impact of the Protein Data Bank on new drug approvals by the US FDA. The first paper, which I co-authored with John Westbrook, that was published in Structure, showed that discovery and development of nearly 90% of the 210 new drugs that were approved by the US FDA from 2010 through beginning of 2010 through the end of 2016 was facilitated by open access to PDB structure data. The impact of these structures ranged all the way from target validation and understanding target biology to structure-guided drug discovery, as you can see here with the structure of BRAF with the small molecule kinase inhibitor vemurafenib bound in the active site. This compound is used to treat late-stage melanomas, which are driven by, in many cases, by mutations in BRAF. And the most interesting feature of this particular story is that the drug was optimized using structure-based methods for the mutant form of the enzyme that causes that's the driver mutation for the melanoma. 50% of patients with late-stage melanoma have a particular characteristic mutation here in the enzyme. And the drug was designed for that 50%, and the drug was approved subject to the results of a diagnostic test, which confirmed that the patient fell into the 50% group that have the mutation and could therefore benefit from the treatment with the drug. The drug is not approved for the other 50% of patients with late-stage melanoma who will get no benefit from the drug and could actually, of course, experience deleterious side effects. Every drug is a double-edged sword. Our second paper in this area, published earlier this year as a cover article in Drug Discovery Today, examined the impact of PDB data and structural biologists on new anti-cancer drugs approved by US FDA from the beginning of 2010 through the end of 2018. So that's 9 years instead of 7 years for the previous study. We showed that discovery and development of approximately 90% of 79 new neoplastic drugs was facilitated by open access to PDB data. And you can see that these new drugs apportion between pills on the left, there are 54 of them, and 25 biologics, almost all of them being monoclonal antibodies, such as the structure depicted here. In most cases, for the small molecule drugs, 70% of the 54 small molecule anti-cancer drugs that were approved 2010 through 2018, we were able to show that they were actually the products of structure-guided drug discovery performed by drug hunting teams in the biopharmaceutical companies. In most cases, these companies benefited from open access to the structures of the target proteins that had been previously contributed by academic structural biologists. In virtually every case for the 79 new anti-cancer drugs, the 3D structure of the target protein was in the PDB more than 10 years before the drug was approved. So it should not come as any surprise to you then that the PDB is widely recognized as being comprehensive, authoritative, and of very high scientific value. It's been certified by a non-governmental organization known as Core Trust Seal and certified as a trustworthy data repository, and is regarded as a gold standard exemplar open access archive for biological data as a public good. And I use the term public good quite deliberately because virtually all of the structures of the target proteins of these 79 new anti-cancer drugs were determined by academic researchers and put into the public domain, put into the PDB. The research that was carried out by these academic researchers to generate this structural information came from taxpayer money, either in the United States or in Canada or Europe or Asia. And this is information, this is research data that was generated with public money, and these data should remain available to everybody with no limitations on usage and at no charge, so that they can be used to promote the public good. So let me give you a little bit more detail now on how PDB data do impact drug hunting and contribute to the public good. 3D structures are vital for understanding the role of proteins in human health and disease, because of course we know that much of the function of a biological macromolecule derives from its form, from its three-dimensional structure. Knowledge of the structures themselves tells us about target druggability, helps us identify potential drug binding sites. Once that's done, co-crystal structures, typically with so-called tool compounds, help us understand the chemotype binding properties of the drug binding sites that have been identified in the target druggability assessment. From there, what typically happens is that company drug hunting teams, which include protein crystallographers, pursue structure-guided drug discovery in which the optimization of the lead series, the chemical lead series for potency and selectivity is being driven by knowledge of 3D structure. And in the most favorable cases, the 3D structures of potential off-targets are also available, so that co-crystal structures with the off-targets with members of the lead series can be analyzed to help ensure that the final compound that goes into animal and ultimately into human testing is in fact as selective as possible. The reason that this is so important is that the bulk of the side effects that we feel when we take any drug are not the result of binding to the target protein. They're the result of unwanted binding to other proteins in our bodies that may cause completely different outcomes, totally unrelated to the mechanism of action of the drug when it binds to the target protein. And then the final way in which 3D structure information can contribute to drug discovery is during to approvals is during the so-called early stage drug development. So I'm talking now about the testing that's done to look at the so-called ADME properties of the small molecule. So absorption, distribution, metabolism, and excretion. And here I'm talking about potential inhibition of cytochrome P450s, potential binding to the human ether-agogo channel, which causes prolongation of the QT interval in the electrocardiogram, and may cause a lethal arrhythmia in a very small number of patients. And finally, for anti-cancer drugs, one of the things we're concerned about is binding to the p-glycoprotein drug resistance pumps, with the anti-cancer drug actually being pumped out of the malignant cells. So having provided that summary, I want to end with what is a very sobering postscript. Nsp5 amino acid sequences are highly conserved across all known coronaviruses. And here I cite the example that the SARS-CoV-2 main protease Nsp5 is 95% identical in sequence to its counterpart in SARS-CoV. The 3D structures of the SARS-CoV-2 Nsp5 and the SARS-CoV Nsp5 are virtually identical to one another. And here's an overlay. The original SARS-CoV-2 Nsp5 is shown in gold, purple, and blue, and with the covalent inhibitor bound here in the active site. And the green ribbon diagram is the structure of PDB-ID 1Q2W. This is the SARS-CoV Nsp5 that my company, SGX Pharmaceuticals, contributed to the PDB, no strings attached, in 2003 at the height of the SARS epidemic. The root mean square deviation on alpha carbons between the two structures is very low. It's smaller than the diameter of one hydrogen atom, coming in at 0.8 angstroms. If we consider only the amino acids that are lining the binding site for the substrate, the active site of the enzyme, the root mean square deviation goes down to less than half of the diameter of the hydrogen atom, 0.5 angstroms. So in 2003, when I was serving as Chief Scientific Officer of SGX Pharmaceuticals, my colleagues and I determined this green structure, and we put it into the PDB immediately. And it was released, no strings attached. It was published in the New York Times. I think it's the only structure in the PDB that's actually been published in the New York Times. And at that point, any drug discovery team anywhere in the world was free to use this information as a starting point for structure-guided drug discovery, which, as I've just shown you, is an extremely effective means of producing enzyme inhibitors that can provide enormous benefit to patients and to their families. Had a decision been made in 2003, 2004 to invest a few $100 million in a SARS-CoV main protease inhibitor, we would have a drug today for SARS-CoV-2, and we would have been in a position to save more than 1 million lives and to prevent trillions of dollars of economic losses worldwide. Unfortunately, that did not happen. The free market in emergency medicines did not work. The free market failed to provide the right financial incentives for one or more biopharmaceutical companies, including my own, to invest in an effective Nsp5 inhibitor. Now, that was 2003, 2004, 2005. In the early teens, MERS crossed the species barrier from camelids to humans, causing a second coronaviral epidemic with many fatalities. And at that point, government, industry, and non-governmental organizations should have come together to prepare for a third coronavirus epidemic. And unfortunately, they did not. Very few countries actually did what was necessary to get ready for another SARS epidemic and to avoid having to shut down their economy and to safeguard the population. So I am passionate in my hope that we can learn the lesson from the current pandemic and avoid the next time that a coronavirus crosses the species barrier, because it's not a question of if, it's just simply a matter of when. Now, those of you who are homeowners know that responsible homeowners purchase fire insurance before the house burns down. We scientists are among the best positioned to influence policymakers to help ensure that we prepare adequately and responsibly for the next coronavirus epidemic. And I hope we do, because, mark my words, there will be another one. So with that, I'll end on a much more positive note and extol the virtues of the very large team of people who stand behind me and support me at the RCSB Protein Data Bank. I'm only able to give this talk because of their talents and their efforts in supporting tens of thousands of depositors worldwide and millions of users worldwide. And I also want to thank those tens of thousands of depositors, which now number nearly 50,000 over the life of the PDB, and the many millions of PDB users who come from every sovereign country that's recognized by the United Nations today. And with that, I will stop sharing, and I will perhaps have time for one question.
Okay, well, there are several questions in the chat. I'll just ask the first one, and then Stephen, perhaps you can answer the remaining on the chat itself.
Sure.
So Sanjay Day asks, does MPro interact with any specific host proteins like PLPro does with ISG15 or ubiquitin and help in viral infection? Why do you think SARS-CoV-2 has two proteases, unlike other RNA viruses where they have only one protease? Does it pose a role in increased virulence?
So that last question is very interesting. I don't know the answer to that question, unfortunately, but I think that's an intriguing thought. As to why the coronaviruses have two proteases, unlike hepatitis C virus, I simply don't, or HIV, I don't know. I don't think that the interactome of the main protease has been as extensively studied as that of PLPro, so I'm not able to answer that question.
There was a paper out from UC San Francisco researchers where they looked at the interactome.
That's right. Yes.
Maybe they found something there.
Perhaps you could put that link into the chat for our colleagues. So Xavi Ruiz asks about whether or not the catalytic His mutated MPro is still active. We really don't know, and I think we need to do some experiments. And I'm hoping that if we don't get to them when we publish this paper, somebody will do the experiment and tell us what the answer is. Jeffrey Yang asked an important question: how similar is the MERS-CoV main protease to SARS and SARS-CoV-2? It's only about 50% identical, but it's virtually identical. I think there's one amino acid difference in the active site. And it may be that in terms of tackling the coronavirus Nsp5s, one might need to make a couple of different, possibly a few different inhibitors covering different clades of Nsp5 across the coronaviruses. We have an enormous amount of information about sequence available now, so that could be done.